So, have you ever held a tiny medicine tablet in your hand and actually wondered how a dusty, clumpy, completely chaotic powder gets perfectly engineered into that sleek, life-saving little pill? I mean, it's not magic, though honestly, it kind of feels like it. It all comes down to the hidden, microscopic physics of small particles. Welcome to our explainer. Today, we're decoding the absolutely fascinating science that makes modern medicine physically possible.
Okay, let's dive into this. The secret to that perfect pill lies in a highly specialized field called micromeritics. This is literally the ultimate scientific solution to the massive physical challenges of manufacturing medication. Basically, micromeritics is the field of science dedicated entirely to the study of small particles, especially powders. It's all about figuring out their size, their shape, their surface area, their porosity, and their flow properties. Because think about it. Without this science, a pharmaceutical factory would just be a room full of unmanageable dust.
Now, you might be wondering, why go to all this trouble? Well, the stakes here are incredibly high. If we don't get the physical math right, the medicine simply won't work in the human body. Period. Do you want the drug to absorb faster? You'll need smaller particles to get a larger surface area, which directly improves how fast it dissolves and enhances its bioavailability. Want to guarantee every single pill has the exact same dose? You've got to have uniform mixing. On the factory side, this science dictates how well the powder flows through machines and controls the stability of liquids, so the drug doesn't just sink to the bottom of the bottle. And of course, if you want your tablet to be hard enough not to just crumble to pieces, you need high compressibility. That is micromeritics at work. So, here's our roadmap for today. First, enter micromeritics. Second, measuring the invisible. Third, separation and settling. Fourth, counting and surface area. Fifth, densities and packing. And finally, number 6, the science of flow. Let's get right into it.
Section 2. Measuring the invisible. Sizing up tiny specks. This brings us to our first really big hurdle. How exactly do you measure a speck of dust, especially when it's not a perfect, round little sphere?
Because real-world pharmaceutical powders, they are highly irregular. They're jagged, they're lumpy, and they are definitely not perfect spheres. So instead of fighting those weird shapes, scientists cleverly measure them by comparing them to an imaginary, perfect sphere. We use what are called equivalent diameters. For instance, the surface diameter is the diameter of a perfectly round sphere that has the exact same surface area as our weirdly shaped particle. The volume diameter equates to a sphere with the same volume. There's also the projected diameter based on the particle's 2D shadow, the Stokes diameter based on how fast it sinks in a liquid, and the sieve diameter based on the size of the hole it can just barely squeeze itself through.
And a frequency distribution curve brilliantly illustrates why those measurements matter so much. If you imagine graphing this out, mapping the particle size against the percentage of particles at that size, you get a curve. You almost never have a powder where every single grain is exactly the same size. So by graphing the particle size distribution, scientists can see the entire range in a sample. Getting a nice, tight bell curve is exactly what ensures our powder mixture is consistently uniform, and that guarantees that every single tablet in a batch is identical. Moving on to part 3, separation and settling. Now that we know how we want to measure these things abstractly, let's look at the actual, real-world lab procedures used to physically separate and size them.
The most straightforward way is the sieve method. Just imagine a stack of metal meshes, kind of like a high-tech flour sifter. You arrange them in a stack with the biggest holes up top and the smallest at the bottom. You dump a known amount of your powder on that top layer, and then you mechanically shake or vibrate the whole stack for a set amount of time. The bigger chunks stay trapped on the higher levels, while the tiny ones fall all the way through to the bottom. Then, you just weigh the powder collected on each tier, and boom, you've found your particle size distribution. It's super practical, but it's really mostly used for larger particles, anything over 38 microns, like standard tablet granules.
But wait, what if your powder is finer than 38 microns? Well, for that, we use the sedimentation method. This usually involves a device called the Andreasen pipette. This whole setup relies purely on gravity and Stokes' law, which essentially says that larger particles settle faster in a liquid, while smaller ones kind of float around and stay suspended longer. So you disperse your superfine powder in a liquid, shake it up, and let it sit. Then at fixed intervals, you use the pipette to draw a sample from a very specific depth. By drawing and weighing those samples over time, you can calculate exactly how fast the particles are falling. And that gives you their Stokes diameter. Pretty clever, right?
Okay, part 4, counting and surface area. We're shifting gears from physical shaking and settling to some really high-tech counting methods. Remember, measuring the outside surface area is absolutely vital for fast drug dissolution in the body.
Let's look at how this builds into even more advanced techniques, like the culture counter method. This is a fully electronic way to count and size particles. So the powder is dispersed in the liquid that conducts electricity, an electrolyte. This liquid is then forced through a microscopic hole. As each individual particle passes through that tiny hole, it physically displaces some of the liquid. And the principle here is brilliantly simple. Because the particle itself doesn't conduct electricity as well as the liquid does, it causes a momentary hiccup or a spike in the electrical resistance. An amplifier records these changes. Every pulse is a counted particle, and the actual size of the pulse tells us the exact size of the particle. It is exceptionally accurate for really fine powders.
Now, if we want to measure surface area directly, we use the air permeability method, typically with an instrument called a Fisher subsieve sizer. This setup packs a known amount of powder tightly into a bed. Then, an air pump pushes air through that packed powder at a totally controlled rate. Think of it like trying to force water through coarse coffee beans versus really fine espresso grounds. The finer the powder, the more friction and resistance it creates. So, by measuring the pressure drop and plugging it into a complex formula called the Kozeny-Kármán equation, scientists calculate the specific surface area of the entire sample based purely on how hard it is to push air through it.
Section 5. Densities and packing. Let's zoom out for a second. We've looked closely at single particles, but what happens at the macro level when millions of these tiny grains are dumped into a container or a massive tablet press?
Well, we have to consider packing arrangements. Literally, how do these particles physically sit next to each other? There are two major extremes to consider. First, cubic, or loose packing. The particles are stacked directly on top of each other, like a cube. This arrangement has a crazy high porosity of about 47.6%. That means almost half the volume of the container is just empty air. It gives you terrible flow, but it's very easy to compress. On the flip side, we have rhombohedral, or tight packing. Here, the particles nestle tightly into the gaps of the layer below them, which drops the porosity down to roughly 26%. This tight packing flows a lot better, but it's significantly harder to compress into a solid tablet.
Now, what's really fascinating here is how those empty air spaces, which we call voids, completely change how we calculate density. In Micromeritics, we basically look at three types of density. First is bulk density, which is the mass of the powder divided by its total volume. It absolutely includes all those empty voids. Second is true density. This completely ignores the air. It's the density of just the solid material itself. And finally, tapped density. This is the density after you physically tap or bang the container, forcing all the particles to settle closer together. Tapped density still includes voids, but they're greatly reduced.
Section 6. The science of flow. Manufacturing the pill. Ultimately, all of these measurements, the size, the shape, the area, the density, they all point toward one massive goal, the science of flow. Because if a powder doesn't flow uniformly, it's game over for the manufacturing process.
The simplest and, frankly, most elegant way to measure how well a powder flows is the angle of repose. If you just slowly pour a powder onto a flat surface, it naturally forms a cone-like pile. The angle formed between the flat horizontal surface and the slope of that pile is your angle of repose. You calculate it just by taking the tangent of the angle, which is the height of the pile divided by its radius. This one single visual measurement tells you so much about the internal friction of the particles. If they are jagged and sticky, they'll stack up high into a really steep cone. But if they're nice and smooth and round, they'll just slide off each other into a flat, wide puddle.
And here is the magic number for the pharmaceutical industry, less than 30 degrees. If the angle of repose is under 30 degrees, it means you have excellent flow. The powder is going to run perfectly smooth through the factory hoppers and perfectly fill every single tablet mold. But if you hit 40 degrees or higher, the flow is poor. And that means the engineering team has to go all the way back to the drawing board and reformulate the powder.
Beyond that angle, we also rely on flowability indices. Remember bulk density and tapped density from a minute ago? Well, by comparing those two numbers, we get what's known as Carr's index and the Hausner ratio. And the best part is, you don't have to do the complex math every single time. Engineers just use these cheat sheet tables. If your Carr's index is between 5 and 15%, or your Hausner ratio is around 1.05, you instantly know your powder flowability is graded as excellent. It's just a fast, universally understood standard to make sure the manufacturing process will run totally perfectly.
And that wraps up our journey into Micromeritics. We've gone all the way from figuring out how to measure weirdly shaped microscopic dust to ensuring that millions of those particles flow like water into a perfect uniform tablet. It's literally a masterclass in physics and engineering, and it's hiding right there in plain sight.
So I'll leave you with this. The next time you take a simple painkiller, will you think about the millions of perfectly measured, beautifully flowing particles engineered inside? The physics of the perfect pill is sitting right there in your medicine cabinet. Thank you so much for joining me on this explainer, and stay curious.